Hello students, once again welcome back to our biology class. Myself Dr. Raghavendra here. Well, in our earlier class we have discussed about some important concepts related with the Human Genome Project. So, Human Genome Project it was ultimately intended to identify number of active genes in the human DNA, and that has been identified nearly 30,000 genes. How it is possible when the bases are somewhere 3 into 10 raised to 9, that much huge number of bases, and ultimately we are able to identify yes the number of genes which are actively coding for the proteins. Well, all it is possible because of the wonderful technique what we called as the DNA fingerprinting, and then a polymerase chain reaction. Okay, then there is a Frederick Sanger's method of automated sequencing. All that had made the Human Genome Project possible. Now, in today's class, we are going to discuss about most important technique what we called as the DNA fingerprinting. Yes, this DNA fingerprinting has number of wide applications. Yes, we are going to study about number of important concepts. What is the methodology? What is the application? What is the significance? What is the basic definition? Also, we are going to study about DNA fingerprinting. So, the DNA fingerprinting is also called as DNA profiling. DNA profiling. So, the another word for DNA profiling or fingerprinting is also called as the DNA typing. Yes, DNA typing. Fine. So, as such, this technique, yes, developed by Alec Jeffrey. So, the scientist Alec Jeffrey developed the technique. So, the technique developed by Alec Jeffrey 1984, when he was studying or sequencing the protein myoglobin. Yes, what is myoglobin? It is the pigment, the red color pigment that is present, the protein present in the muscles. So in the blood we have the hemoglobin like that. We can find the myoglobin in the muscles. He was studying the sequencing of the myoglobin. At that time he developed this technique what we call as DNA fingerprinting or DNA profiling. So as such here in the DNA fingerprinting or profiling, what we can be mainly studying. So all humans have the DNA which is 99.9% similar to one another. So what is then? The difference is 0.1%. So that variation, yes, we are able to understand and identify mainly by using the technique what we call as the DNA profiling or DNA fingerprinting. As such, human genome, we can say that human genome has 3 into 10 raised to 9 base pairs. These many huge number of base pairs, and among that we are going to identify that variation, that difference from person to person. So the technique, yes, the technique extensively helpful to resolve parentage disputes as well as crime cases, and also helpful in evolutionary studies, and yes, mainly used in bioarchaeology, we can say bioarchaeology. Yes. So from the fossils, yes, we can isolate the DNA, and then that DNA and human DNA can be compared and we can identify the phylogenetic relations also. Like that, DNA fingerprinting has a number of applications. Yes, the samples can be isolated from the hair, from the nails, from the skin, then salivary sample, sperm sample, yes. And even from the blood, we can say, from the bone, we can say the DNA isolated, and then compare the DNA binding pattern. So then we can resolve the cases such as parental disputes. So the mother will tell that this child is born to so and so fellow, but he is denying. But when we compare the fingerprint, yes, at least 50% of the banding pattern will be similar to that of the father, and 50% DNA banding pattern similar to mother. So in such cases, DNA fingerprinting is very much helpful. From a crime spot, yes, we find out the DNA and compare that DNA with the suspected person's DNA. Yes, we can identify whether the suspected person is a criminal or not. And then from the rape cases, yes, from the vagina, you can isolate the samples of the semen and then compare that sperm DNA sample with the suspected person. Yes, you can identify the criminal there also. Like that, in resolving number of important cases, identifying the phylogenetic relations, resolving the parental disputes, this technique is a boon, we can say. It has been helpful in number of ways. So, what are the important things? Yes, we are going to discuss under the concept what we say DNA fingerprinting as such. We will be discussing more about mainly, let me tell you, definition. Number 2, we are going to discuss about significance. Number 3, we are going to discuss about the procedure of DNA fingerprinting, and we will be discussing about applications of the DNA fingerprinting. Well, so let us understand the basic definition of DNA fingerprinting. So let me tell you, yes. So DNA fingerprinting a technique of identifying similarities and differences between DNA, okay, DNA of, yes, different organisms and human. So 99%, yes, DNA or the sequences or the nucleotide base pattern is very much similar, but 0.01% or 0.1% is mainly the difference. So this DNA fingerprint, mainly we can say that it is based on yes, mainly the variations in the DNA, or we call them as repetitive sequences. So they are also called as satellite sequences. Satellite sequence detection. Okay. So the satellite sequences are very, very unique. Repetition of these sequences are very, very unique. They differ from person to person. So repetitive sequences or satellite sequences, we can say that. Yes. So they are unique and differ person to person. Differ person to person. But one important thing, satellite sequences, no doubt the repetitions, but these repetitive sequences 50% can match with father and 50% with the mother. So that helps in identifying mainly the parentage. Well, so that's, I hope you got an idea very clearly what is DNA fingerprinting. It is also called as DNA profiling. It is also called as DNA typing, we can say. The technique is developed by Alex Jeffrey, okay, when he was studying about a protein called as myoglobin. And then all the human have the DNA 99.9% very similar to one another. The human genome has 3 to 10 raised to 9 base pairs. A huge number of what you can say, mainly the base pairs. Among that we have to identify what is that 0.1% difference. So 0.1% difference is due to the repetitive sequences or satellite sequences. They are non-coding sequence, we can say. Okay. So the technique is extensively helpful to resolve the parentage disputes, crime cases, and used in bioarchaeology and even detection of phylogenetic relations also, we can say. So we will be discussing about their significance also, and then the procedure and applications also. So by definition, I hope you got a clarity. It is a technique of identifying the similarities and dissimilarities or differences between the DNA of different organisms and the humans or the samples, we can say. So how actually it is getting mainly detected? It is getting detected by identifying the satellite sequences or the sequences which are unique to person. They differ from person to person, that we are following. The satellite sequences are basically repetitive sequences. They keep on repeating. So such repetitive sequences we are going to target by using the technique what we call as a DNA fingerprinting. Well, let us understand now mainly the significance of this what we can say, the DNA fingerprinting first. So we will discuss about the significance of DNA fingerprinting, and then we will discuss about the procedure as well as the applications also. Fine. When we are talking about what is the significance, what is the importance, how actually the samples have been isolated, okay. So what is ultimately the use or importance? Yes, we are going to study under the subtitle significance of DNA fingerprinting. Well, so let us go ahead here. Mainly significance. So to carry over the DNA fingerprinting, we need the samples. So sample collection, sample or the DNA, okay, required, isolated from any kind of cell, but most importantly it can be from the hairs, okay, it can be from the bone or osteocyte, we can say, it can be from the blood, mainly the WBCs. Well, then it can be from mainly the saliva with buccal epithelial cells, buccal epithelial cells. The nucleus is there, then only we can isolate the DNA. Then there is even from the skin cells, DNA can be isolated, even from the sperms of the semen sample. Okay, so these are the different sources of DNA, from where we can isolate the DNA. The sample collection will be done. So the significance, as such we are discussing the sample collection, we need to know. Then, so when we talk about mainly these, what we can say, the significance of sample collection, it is done. Now we need to understand mainly the sequencing. How these mainly the what we can say, the sequencing is done. Sequencing, as such, the sequences of the DNA by cutting the DNA using restriction endonuclease enzymes. They cut the DNA at a particular or specific sequences. Okay. So thus the restriction endonuclease enzymes are used mainly to cut the DNA. And then, so when we isolate the DNA, we are cutting the DNA, yes, because the DNA as what we mainly isolate, it is having coding, non-coding, it is having mainly repetitive as well as yes, non-repetitive sequences. So sampling is very very important. Sequencing, how we do mainly the DNA once we are interested to isolate. So imagine there is a method sequencing by cutting the restriction REN means the cells from which the DNA isolated, then what we do, yes, the DNA taken into, so here what we say mainly the DNA isolated by method called as a gradient centrifugation. A technique is applied. Then what we find? We find the bulk DNA in which we are not much interested. It is 90%, but the 10%, what we call it as mainly, yes, the 10% DNA has satellite DNA, we can say, satellite or repetitive sequence DNA, which forms 10%. This is the matter of interest for us. We need to use the satellite DNA when we do number of techniques. It may be the high performance liquid chromatography technique is there, when we use our spectroscopy technique is there, right? So in a mass spectroscopy, yes, it gives a DNA profiling as such. It forms a large peak, it forms a narrow peak, a small peak. So this is called as mainly the bulk DNA, which is of not interested for us, because it may be having variety of sequences, but we are targeting for the repetitive sequences or satellite sequences. So the bulk DNA, and then this is what we say the satellite sequences. So which is a 10%, this is 90%. So we are more interested in satellite sequence, a narrow peak and the large peak of no interest, whereas the narrow peak, yes, which is mainly required for us. This is what we can say a spectroscopy graph of the DNA sample what we isolate. So the sequencing, yes, we do this when we add mainly the particular restriction endonuclease. They cut the DNA at a repetitive sequences only, and that we call as mainly the, yes. So how actually we can classify these DNA? So yes, sequencing is the second, and yes, and sample isolation is the second step or the significance what we are studying. Now the third important point, classification of DNA. Yes, the classification of DNA, it is not like that of the RNA, what we have mRNA, tRNA, no. Here the DNA we are mainly classifying based on the sequences as such. We call it as mainly the bulk DNA which is of not our interest. Then satellite sequences, or we call them as the repetitive sequences. So these are very much unique. Yes, they are very very less percentage also, but they are very very essential for us. So these repetitive sequences, what they are? Repetitive sequences like that here. So CCAA, CCAA, CCAA means on a DNA. Yes. So these are the repetitive sequences what we can find. These are the repetitive sequences. So pattern of repetition is very specific or unique. It repetition pattern differs from person to person. This kind of repetitive sequence we call them as a satellite sequence. So satellite sequences are of two types. So satellite sequences are of two types. One is called as a micro satellite. So they are mainly 2 to 6 base pairs. So these are mainly repetition like this. Here only 5 sequences are repeating. This is what we say microsatellite sequence. And then mini satellite sequence. Mini satellite sequence, which are 10 to 60 base pairs. So thus the repetitive sequences can vary in their length. They may be microsatellite, they may be mini satellite. Our target is micro or mini satellite only, right? And the repetitive sequences are very much unique, and these repetitive sequences are non-coding. So non-coding. Sometime these are also called as VNTRs, means these are also called as VNTR. What is this VNTR? It is also known as variable number of tandem repeats. Tandem repeats. Okay, so that is what we say VNTR. So our target is mainly VNTRs, means classification of the DNA as a bulk DNA and satellite DNA. Yes, in that we find lots of these micro and mini satellites, and they are also called as VNTR. And Alec Jeffrey has mainly focused on these VNTRs and then made the DNA fingerprinting technique possible. So I hope you got a clarity regarding mainly what we say significance as a sample collection, as well as the sequencing by using the restriction endonuclease enzymes. If a cell, we are isolating the DNA, then we are applying a gradient centrifugation method, and when we rotate that whatever the content under a centrifugal force, we find the settlement of the DNA at a different layer, and we find 90% is a bulk DNA, and yes, satellite DNA also we can find over there. The 10% is satellite DNA, that is our interest, because restriction endonuclease, when we cut the DNA, the smaller, yes, repetitive sequences are light, and then they will float and easily they can be collected. So thus the DNA can be bulk DNA or satellite DNA. In satellite DNA we find micro and mini satellites. So we call it as mainly polymorphism. Yes. So DNA with the different forms, we call it as DNA polymorphism. How this DNA polymorphism can do exist? So let me tell you here DNA polymorphism. So the fourth significance is DNA polymorphism. Polymorphism, or we can say that even allelic variations. Allelic variations exist. Yes, more than 0.01, okay, percent we can say. Yes. So almost when 99.9% DNA is similar in all the human, then DNA polymorphism, yes, that is possible, more than 0.01% only. Well, so this kind of DNA polymorphism is due to, yes, DNA polymorphism is due to, yes, satellite DNA, okay, or we can say that due to the VNTR. So VNTR targeted by using restriction endonucleases. Yes, restriction endonucleases. restriction endonucleases, we call them as mainly REN. REN target palindromic or a type of repetitive sequences only, palindromic sequences. Simple example I will tell you. GAATTC, GAATT and C. So this is a DNA, means a EcoR1 enzyme is there. Yes, it can cut the DNA at this point. So EcoR1 is the REN. Okay, it is a type of REN which cuts the DNA at these sequences. Okay, so it is a 5 dash, it is a 3 dash, it is 5 dash, and here it is a 3 dash. So like this it can cut. So thus we are using mainly these variety of mainly what you can say the restriction endonucleases, and we are identifying the DNA polymorphism and cutting the DNA, yes, mainly by targeting the satellite sequences. Now we need to understand, yes, mainly the variation, why it has happened. Variation happened due to the mutations. So mutations are the reason for the variations, and mutations keep on, yes, transmitting generations to generation, and thus they are the reason for mainly what we can say that expression of the variations. So sometime the particular or peculiar kind of variation are expressed generation after generation. So we need to remember this also. Mutations change in DNA, and it is inheritable also. The causes for or mutations can be due to, it can be due to, yes, we can say that sequence either deletion, okay, or it may be insertion. It can be due to transition. It can be due to transversions. So thus mutations can happen because of these many reasons, and we can say that mutations bring variations, and inherited as well expressed. Generation after generation after generation. Yes, there may be number of reasons. We have studied mutagens also. It may be physical, chemical as well as biological mutagens. But at the time of, yes, when the cell is dividing, cell is copying its DNA, that time also there is a high chance of unwanted deletion, unwanted insertion. There may be, we can say that point mutation means one sequence changes or mainly exchange its position with another. That time also the point mutation such as transition means adenine as well as guanine, they are changing their position. Cytosine or thymine changing their position. Transition, transversion, adenine with the thymine, guanine with cytosine may be changing their position. So this kind of changes will bring the mutation. So mutations also, yes, keep on happening every now and then. They bring the changes and they are expressed. So mutations are essential for, mutations bring about evolution, variations as well as speciation, formation of the new species. All these are again part of DNA fingerprinting. Yes, when we analyzing the DNA fingerprint, we will be understanding what is variation, what is evolution, what is speciation, development of the new species. So thus, the mutations are the reasons for the polymorphism also. Yes, mutations also reason for, mainly we can say that for polymorphism, population variation, okay, and thus these are the most important thing, yes, what we have discussed, and we will understand again regarding mainly the mutations as such we have discussed. Remaining point as such, mainly significance is done. We will study about the what you can say procedure as well as applications of the DNA fingerprint in the next class. Till then, take care, study well. Thank you very much.